The most precise gene editing tool known to science originated as an ancient defense mechanism. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, functions as an adaptive immune system within prokaryotes, specifically bacteria and archaea. This system provides acquired resistance against invading bacteriophages and plasmids. The CRISPR locus integrates short segments of foreign DNA, known as protospacers, into the host genome as spacer sequences. These spacers are transcribed into precursor CRISPR RNA, or pre-CRISPR RNA. The pre-CRISPR RNA is subsequently processed into mature CRISPR RNA. This mature RNA guides effector complexes to target and neutralize specific mobile genetic elements. This natural defense mechanism forms the foundational molecular architecture for the engineered gene editing technology. The engineered CRISPR-Cas9 system is primarily derived from the bacterium Streptococcus pyogenes, utilizing the enzyme SP-Cas9. Targeted cleavage requires two essential components. The first component is the Cas9 endonuclease, a large protein responsible for executing the DNA cutting action. The second component is the single guide RNA, or sgRNA. The sgRNA is an artificial fusion of the natural CRISPR RNA and the trans-activating CRISPR RNA, known as tracRNA. Crucially, the sgRNA contains a 20-nucleotide sequence that is complementary to the desired genomic target site. This specific complementarity dictates the absolute specificity of the Cas9 enzyme, directing it to the precise location within the genome for editing. Target recognition is initiated by the Protospacer Adjacent Motif, or PAM. For the widely used SP-Cas9 enzyme, the canonical PAM sequence is NGG, located immediately downstream of the target protospacer sequence. Cas9 must first bind specifically to this PAM sequence to initiate the local unwinding of the DNA helix. PAM recognition facilitates the hybridization of the single guide RNA to the complementary target strand of the DNA. This binding process forms a characteristic R-loop structure. In the R-loop, the single guide RNA is fully paired with the target strand, while the non-target strand of the DNA helix is displaced and looped out. Full R-loop formation confirms the correct target sequence alignment, signaling the enzyme to proceed with cleavage. Once the R-loop is fully stabilized, Cas9 executes the cleavage using its two distinct nuclease domains. The HNH domain is responsible for cleaving the DNA strand that is complementary to the single guide RNA. Simultaneously, the RuvC domain cleaves the non-complementary DNA strand. This coordinated action generates a blunt-ended double-strand break, or DSB, precisely 3 base pairs upstream of the Protospacer Adjacent Motif sequence. The generation of the double-strand break is the critical event that triggers the cell's endogenous DNA repair machinery. The subsequent repair pathway determines the final editing outcome. The precision of the double-strand break location is fixed relative to both the PAM and the guide sequence. The primary cellular response to a double-strand break is the non-homologous end joining pathway, or NHEJ. Non-homologous end joining is an error-prone repair mechanism that rapidly ligates the broken DNA ends without requiring a homologous template. This process frequently introduces small insertions or deletions known as indels directly at the cleavage site. If the size of the indel is not a multiple of 3 nucleotides, it causes a frameshift mutation in the coding sequence. Frameshift mutations typically result in the introduction of a premature stop codon. This leads to the production of a truncated, non-functional protein, which effectively achieves a gene knockout. For achieving precise gene editing, the homology-directed repair pathway, or HDR, must be utilized. Homology-directed repair is active predominantly during the S and G2 phases of the cell cycle when a sister chromatid is available. This pathway requires the introduction of an exogenous DNA template, known as the donor template. The donor template contains the desired sequence flanked by homology arms. These homology arms must match the genomic sequence immediately adjacent to the double-strand break. The cell uses this donor template as a blueprint to repair the break. This mechanism allows for the precise insertion of new sequences, the correction of single-point mutations, or the substitution of large DNA segments. CRISPR technology has rapidly evolved beyond generating simple double-strand breaks. Catalytically inactive Cas9, known as dCas9, retains its DNA binding capability but completely lacks nuclease activity. dCas9 can be fused to various effector domains, such as transcriptional activators or repressors, to modulate specific gene expression, a technique known as CRISPR activation or CRISPR interference. Base editors, or BEs, represent another major advancement. Base editors fuse dCas9 or a nickase Cas9, nCas9, with a deaminase enzyme. Base editors enable the direct conversion of one base pair to another, for example, converting cytosine guanine to thymine adenine, without creating a double-strand break. This modification significantly reduces the reliance on the error-prone non-homologous end joining pathway. Effective delivery of the CRISPR components into target cells is crucial for both therapeutic and agricultural applications. Components can be delivered as DNA plasmids, messenger RNA, or as a pre-assembled ribonucleic acid complex, or RNP. Viral vectors such as adeno-associated virus and lentivirus are frequently used for stable long-term expression in vivo. Non-viral methods, including electroporation and lipid nanoparticles, are often preferred for transient expression and reduced immunogenicity. Applications include treating severe genetic disorders like sickle cell disease, developing disease-resistant crops in agriculture, and engineering microbial pathways for advanced industrial biotechnology processes. A primary technical challenge in CRISPR implementation is minimizing off-target editing. Off-target editing occurs when Cas9 cleaves sequences that are similar but not identical to the intended target site. This activity is often caused by mismatches in the single guide RNA sequence, particularly those located far from the Protospacer Adjacent Motif. Strategies to enhance specificity include optimizing single guide RNA design to avoid highly repetitive genomic sequences. High-fidelity Cas9 variants, such as SP-Cas9-THF1, have been engineered to require stricter complementarity for successful cleavage. Delivering Cas9 as a ribonucleic acid complex ensures rapid degradation, thereby limiting the total time available for unintended cleavage events.
Gene editing therapies are subject to rigorous regulatory review by bodies such as the Food and Drug Administration and the European Medicines Agency, focusing intensely on safety and efficacy data. Ethical considerations differentiate between somatic cell editing, which affects only the treated individual, and germline editing, which introduces heritable changes into future generations. Future research focuses on expanding the molecular toolkit with novel Cas enzymes. Examples include Cas12a, also known as CPF1, and the RNA-targeting enzyme Cas13, which recognize different protospacer adjacent motifs and target substrates. Optimization of in vivo delivery systems, particularly for non-viral methods, remains a key area for advancing clinical translation.